Um, Hello everyone. Want to greet you and welcome you to this lesson. It's an introduction to nutrition. By the end of this topic, you should be able to do the following. I hope you will pause, analyze whatever is written there, so that by the end of this lesson, you can at least achieve 100% of what is there, because everything that we are going to look at is really simple and basic. Nutrition as itself, I would define it as the way in which organisms obtain food. How a rat obtains food. How does a plant obtain food? How does a bedbug obtain food? How does a mushroom obtain food? How does an amoeba obtain food? How does bacteria obtain food? All those questions can be answered by what we call the biology of nutrition. So the various way that living organisms can acquire food is what we call nutrition. But the food we are talking about, from our primary, we know that food, we eat it for primarily two reasons. One, to get energy. Two, to grow. So when you eat food, that food must be able to enable you to grow, and it must also enable you to get energy. So whatever is, like, for you to grow, you must synthesize. Your body must make new structures, new cells, new tissues, new what. So the matter that it uses is what we call the materials, and that those materials come from the food you eat. That's why without eating you can't grow well. Even when you eat, you don't eat very well. That means you also don't grow very well. Secondly, that food must contain energy. That energy is always contained in the chemical bonds, the covalent bonds of the so-called carbohydrates, proteins, and even lipids. You all know that from our chemistry, when bonds are forming, energy is released, and when bonds are being broken, energy is what is absorbed. So through those breakage and bond formations, we get what we call energy. Energy is released, and it can power us to do our activities. So those two qualities must be possessed by food. That is energy and the materials. That materials used for building of our structures like tissues. So organisms are very diverse in nature. They are very different from each other. Imagine, look at yourself and then think about the bacteria, because you can't see the bacteria, but at least you can think about it. Think about the euglena in the pond. Think about amoeba. Think about algae. Think about so many things. We are really different, but we all must obtain food in order to survive. So nutritionally, we categorize them into two groups. We have those ones that can make their own food, and those ones that cannot make their own food. So those organisms that can make their own food, we call them autotrophs. Those ones that depend on others, are called heterotrophs. The word hetero means, basically, it means different, or it may mean other, something else, others. So heterotroph, the word troph means feeding. So heterotrophs, that means you are either feeding on others or you are obtaining your food from others. For example, all animals are heterotrophs. All fungi are heterotrophs. Some protists are heterotrophs, and also some bacteria, they are also heterotrophs. However, plants are majorly autotrophs. Some bacteria are autotrophs, and some protists are also autotrophs. So, how is the difference? However, like, both must obtain something. You can't just make food out of the blue. So even these autotrophs, at least they must get carbon dioxide from the environment and then water, and then either they can use water or hydrogen sulfide depending on the organism to make the food. However, the difference is that these guys, for them, they use simple inorganic compounds. When you look at carbon dioxide, really, it is only one carbon atom and two oxygen atoms. When you look at water, only two hydrogen atoms and one oxygen atom. But now when you look at a carbohydrate, for example, starch, it is made up of thousands and thousands of carbon atoms. It is made up of thousands and thousands of hydrogen atoms. It is made up of thousands and thousands of oxygen atoms. That means heterotrophs, for them, they get already complex organic compounds from autotrophs, of which autotrophs, for them, they get the simplest inorganic compounds, and then they build them to make them complex. So when they build them, they can use some of them for their own purposes, and then others will be eaten by the heterotrophs. Types of autotrophic nutrition. Basically, we are going to focus our discussion today on autotrophic nutrition. Autotrophic nutrition, as we have said, for them, it involves getting simple inorganic compounds and build them to make them complex. So, however, we have different types. Or we can do that. We can build these complex organic compounds from the simple inorganic compounds using different approaches. One of the approaches is to use energy from chemical reactions. You know, as I told you from chemistry, when chemicals react, there will always be an energy change. Either energy is released or energy is absorbed. So when energy is released into the environment, these organisms, mostly bacteria, they can harvest or harness this energy and use it to power or to drive the processes which will lead to the formation of food from simple carbon dioxide and water or hydrogen sulfide. Then, when it comes to plants, some protists, for example, the algae, and some other bacteria, for them, they use energy from the sun. This energy, it is in form of solar radiation. They trap it, of course, usually using a certain pigment. We shall see later those pigments. It can be chlorophyll, it can be what we call the carotenoids. We shall see them. They trap this solar radiation. Then the energy of the radiation is used now to drive or to power the processes that will lead to the formation of of this complex food. Hope we are at par. So in our discussion still, we are going to focus mainly on photosynthesis. I want you to note that this is among the most important things in the world or on earth. It is the one that sustains millions and millions of people. Recently, we are about to clock 8 billion people. But those 8 billion people are supposed to be supported by photosynthesis. So it is of great importance that every biologist is exposed to this great thing. From your primary, you know what photosynthesis is. However, I want to highlight a few things here. As you have seen, from primary, we know that the photosynthesis is a process by which green plants make their own food. Is it? Very good. So they stop there. But I want you to note that it's not only plants that make their own food. I've told you even protists can make their own food. Even bacteria can make their own food, right? I've told you also that even some other plants are not even green, but still they can photosynthesize. Are we together? Yes. So that definition is not wrong. It is correct for those children, but for you, I don't expect you to do that. Any organism can carry out photosynthesis as long as it has the necessary pigments to trap the solar energy, and as long as it has the capacity to fix the carbon dioxide and water or hydrogen what? Sulfide. It's not just plants, it's not just green plants. It can be any organism, ranging from bacteria to the plants themselves. However, animals do not photosynthesize. Even fungi also do not photosynthesize. Another thing is that you must mention the source of energy. When it comes to photosynthesis, the sun is the source of what? Energy. Actually, the word photo means light. So the sun is the major source of light in the universe. And this energy comes from the sun. That by you specifying the source of energy, you are differentiating between photosynthesis and chemosynthesis. Well, I've just told you about the significance of photosynthesis. I told you we are basically depending on this photosynthesis. But how do we depend on it? It is the major source of food to all organisms on Earth. And I told you food has two qualities. It must contain energy in terms that is chemical energy, the energy which is found in the energy which we find in chemical bonds. We call it chemical energy. Then it must be able to be used to build our tissues, our cells, so that organisms can grow. So that is materials. Even synthesizing other substances in our body. For example, you need to synthesize some hormones. You need to make some hormones in your body. You need to make a lot of things. Even saliva itself, you secreting saliva, mucus, so many things. All those ones, you synthesize them or you make them. Your body makes them from the food you eat. That's why if you eat poorly, definitely you find that your body will begin to misbehave. Some girls may even fail to get pregnant because they can't synthesize enough eggs. Maybe when they are not eating well. You should eat well so that you get the energy to do all the activities that you need. Some of you doze in class, you sleep in class because of the food you eat. You eat poor quality food, definitely you have to sleep in class because the energy you are getting can't really support a human being who's willing to study. And some of you have become dwarfs because of eating non-nutritious food. So why? You should focus on these two things. Food must be able to give you energy and materials for the functions that we mentioned above. Then photosynthesis also is a major source of oxygen. Are we together? That one, I don't want to overdwell on it, but you know that at least from your primary, the byproduct of photosynthesis is oxygen, and it is from this photosynthesis that we get all the oxygen that we breathe, the oxygen that is sustaining the earth. And for you guys, I don't know how you see plants, but plants love us so much, can do all this stuff for us. Minimizes effects of global warming. So what is this global warming? Well, just to give you a small glimpse, when we release carbon dioxide in the atmosphere, for example, when you burn fuel, when you burn charcoal, you burn bush, all these things are organic in nature. They are made up of carbon. So when you burn them, technically what you are forming is carbon dioxide. This carbon dioxide escapes into the atmosphere and forms a layer or a blanket around the earth. So now when this solar radiation comes, at first when it's just coming, it has strong radiations, it has high energy, so it has the capacity to penetrate through this blanket, and then it will hit the earth. So the surface of the earth will reflect this radiation. Now, when this radiation tries to escape back into space, it finds the other blanket. This time around, the reflection is of a low energy. It's not as strong as the one that came. The incident radiation was technically had high energy compared to the reflected radiation, so it cannot penetrate through the what? The other blanket of carbon dioxide around the Earth. So since it is trapped in between the other blanket and the Earth, that energy increases the temperature of the surface of the Earth. And now gradually the temperature of the Earth begins to uh, rise and rise and rise. So eventually you find that we have what we call ice caps. For example, in Antarctica, much of the land is covered by ice. So when the temperatures increase, that ice begins to melt. When the ice melts, find that water now begins to flood. The oceans begin to flood, and that's why you hear that in India, floods are killing them. In the Philippines, floods are killing them, etc., etc., etc. Because they are near the other mountain, they are the Himalayas, those mountains which are covered by snow. So when you find that when the temperatures now increase, technically the snow will melt and the water volume will increase, and now it will begin flooding. So those people will die. That's one. Two, this global warming itself has there are some organisms which cannot tolerate increased temperatures, which are more protected by low temperatures. These can be plants and some animals. So if you increase the temperature beyond the normal, that means they will begin to die out slowly by slowly. So that means extinction will occur. Extinction is the death of all organisms of the species. For example, if all human beings die, we shall say that human beings they became extinct. In case I don't know who will say that, but maybe there will be some other organism which can say that. So they will say that human beings became extinct. The way we can say that dinosaurs became what? Extinct. So you can see what global warming can do. But this one can be prevented if we allow photosynthesis to take place. And how do we do that? By making sure that we protect the trees which are there, and also planting new ones, and replanting areas where trees have been cut down. Hope you know that as a biologist. So that what we have talked about, basically, I think every Tom, Dick, and Harry knows. But I want us now to slowly enter the biology of photosynthesis. And I want us to first look at where exactly does this photosynthesis take place. First of all, I told you photosynthesis takes place in plants, some bacteria, some protists like Euglena, algae, etc., etc., etc. So many organisms. Now, in plants, photosynthesis will take place in virtually all green parts. Every green part you see, just know photosynthesis takes place there. These can be leaves, these can be stems, these can be roots, some, every part where you can see something green. Are together? So that means wherever you see anything green, it is always chloroplasts. They will have chloroplasts there. And these chloroplasts contain what we call a pigment we call chlorophyll. So this chlorophyll is found in chloroplasts. So since what makes it green is that it will absorb, you see, when we have what we call the visible light. The visible light is this section of the radiation that comes from the sun, which we can see. And this visible light contains so many colors. Each color of light represents a certain wavelength. For example, we have red light, we have blue light, we have green light, we have yellow light, we have orange light, we have violet light, we have ultraviolet, of which ultraviolet we can't see it. We also have infrared. Infrared we also can't see it. So at one end we have infrared, at the other end we have violet light. I mean ultraviolet, we can't see any of them, but we can see violet and we can see red. So, however, from your physics, you know that from your physics, you know that a pigment will absorb all other colors, and whatever it will reflect, that is how that pigment will appear. For example, if you shine light on a black object, all the light will be absorbed and nothing will be reflected. That means you'll see that thing as dark, so it will be black in your eyes, and your brain will interpret it as what? Black, because it's not reflecting any light to you. If you get something white and you strike it with light, it will reflect all the colors, it will not absorb any color. So when all the other colors, when red, green, blue, yellow, orange, violet, when all of them, when they combine, they form white. I hope you know that one from physics. So now, when these chlorophylls, when you strike them with visible light, that I mean white light, which contains all the other colors, it absorbs all the other colors and it it reflects green color. So that means it does not absorb green color. So that's why we see these guys as green. Hope you've understood that. So that is in plants. Now, these chloroplasts, I want you to know that they are found inside the cells of plants. Now, if you compare a photosynthetic bacteria and a chloroplast, they are almost equal in size. Hope you are getting me. That means if they are equal in size, that means a bacteria, a photosynthetic bacteria, cannot have chloroplasts inside its cells, inside itself, because they are unicellular. So a cell of a bacterium cannot contain a chloroplast because the bacteria and the chloroplast, they are almost of equal size. So now, where does photosynthesis take place in bacteria? Well, the cell membrane or the plasma membrane of the bacteria is folded in some regions. It forms folding. So it folds, and then when it folds, it will that where it has folded, it will form where that will be the site where photosynthesis will take place. I hope you've got it. The plasma membrane or the cell membrane of bacteria folds in some regions to form what we call photosynthetic membranes. Bacteria do not have chloroplasts. Why? Because bacteria and chloroplasts, they are almost of the same size. Now, in protists, for example, algae, for them they have chloroplasts. However, their structure is different from that one of plants. Why? For example, if you look at the algae which we call Spirogyra has spiral chloroplasts. I don't know if you know the structure of DNA. That's how these guys are. The chloroplasts of Spirogyra, they are also in that kind of shape. They form a spiral. That's the reason why even the organism is called Spirogyra itself. So basically we are going to concentrate on plants. That is the structure of a chloroplast. You can see it has two membranes, the outer membrane and the inner membrane. I want you to know that these membranes look almost like the plasma membrane of the cell. And actually there is a hypothesis that says that, these chloroplasts were once photosynthetic bacteria which were engulfed or swallowed, if I'm to use an English word which you understand very well, by a certain amoeba-like cell. So when it swallowed it or when it engulfed it, this amoeba-like cell failed to digest to digest the photosynthetic bacteria, and now they became they developed a mutualistic relationship. From ecology, you know, a mutualistic relationship is when organisms live in a certain relationship and each of them benefits. For example, this guy could photosynthesize, produce the oxygen, and then which could be used by the other organism, and even photosynthesize and produce the food which the other organism could use without having to move around looking for food and stuff. And then the other organism in turn would give this guy, the amoeba-like organism would give the photosynthetic bacteria, it would give it protection from the environment. So at the end of the day, they would both survive well, compared to when the other compared to if each of them stayed separately. So that is called the endosymbiont theory, trying to explain where chloroplasts came from and stuff. So this is the structure of the chloroplast. It has two membranes. So it is assumed that the inner membrane was the original membrane of the bacteria, of the photosynthetic bacteria. And now during the process of engulfing, you know, you must form a food vacuole, like part of the plasma membrane of the amoeba-like organism had to break off to surround this newly engulfed particle to form what we call a food vacuole, right? So, however, since it failed to digest it, so this now that's how this guy now got two. That's how it got two membranes, of which both of them are similar membrane. Then we have what we call the intermembrane space. That is the space between the two membranes. And then we shall have these guys that you are seeing inside. They are called thylakoids. Thylakoids. That's the SP, thylakoid. If it is one, it is a thylakoid. If there are many, they are thylakoids. However, in regions where these thylakoids meet, they form like stacks of coin. If you have ever piled coins to form something, a vertical stack of coins, 500 coins, 500 coins. Those stacks, you can see them here, these bundles. We call them grana. If there are many here, if it is one, like the way it is printed here, it is called a granum. Granum. If there are many, you call them grana. Now, apart from that, we have what we call the stroma. This stroma is similar to the cytoplasm of the real cell. So it is assumed that it was originally the cytoplasm of the photosynthetic bacteria. So it contains DNA, saturated DNA, like the cytoplasm of bacteria. It contains ribosomes, the 70S ribosomes. It contains some enzymes. It contains a lot of substances that are found in the real cytoplasm of other types of bacteria. So that is the structure of the chloroplast for you. So they are biconvex in shape. Biconvex means, as you have seen here, this shape is called biconvex. If you can remember well the structure, the shape of a red blood cell, they describe it as being biconcave. So this is biconvex. They are double membraned, inner and outer membranes. I've explained where they come from. There is that intermembrane space. There is a system of flattened membranes known as thylakoids, whose structure is similar to that of the plasma membrane. Still, this is the membrane of the thylakoids. You know, the thylakoids are basically flattened membranes. Like you get a membrane, you flatten it, you form those things. So, and its structure is similar to that of the plasma membrane. And then we also have the granum. where we have stacks of the thylakoids. If it is one, then grana, plural. Then we have the stroma, I said, which contains ribosomes, the 70S ribosomes, enzymes, proteins, DNA, RNA. I want to clarify more on this case of ribosomes. Eukaryotic organisms, that means those were the from protists living out the bacteria, from protists, fungi, plants and animals, we are called eukaryotes. We have a true nucleus. And also we have our ribosomes are 80S, not 70S. Our ribosomes are bigger compared to the ribosomes of what? Of the protists. Are we together? So that's why now it's funny if you find in a chloroplast, because a chloroplast is found in plants and protists, right? And these are eukaryotic organisms, but their chloroplasts contain 70S ribosomes and the circular DNA. That means all these characteristics are for protists, yet these chloroplasts are found in eukaryotes. That means at one time a eukaryote had to swallow or engulf, as I've explained, the what? As a photosynthetic what? A photosynthetic bacteria. That's why there are these characters. This is evidence that to prove that. And also, I want you to transfer the same explanation when it will come to respiration. If you look at mitochondria, it has also 70S ribosomes, it has circular DNA, it has double membranes, etc, etc, etc. However, for it doesn't have thylakoids, because thylakoids, we shall see them we shall see their role when it comes to the processes, the phases of photosynthesis. Now, the thylakoids, that's where we find exactly the location of what? Photosynthetic pigments. It can be chlorophyll, but we also have other photosynthetic pigments, and we are going to look at them shortly. So types of photosynthetic pigments. We have chlorophyll A, B, and C. Now, all photosynthetic organisms must have chlorophyll A. Then some organisms have chlorophyll A and B. Then others have A and C. But as a must, they must have what? A. Chlorophyll A must be there. Then it can be given chlorophyll A, B, or chlorophyll A and C. Then we also have what we call carotenoids. In plants, in green plants, we have mainly chlorophyll A and B. Then in algae, we can have chlorophyll A and C. We can also have chlorophyll A and B, depending on the type of species. Then we have carotenoids. So what are these carotenoids? Carotenoids, they give color to the structures of the plants. For example, fruits. For example, old leaves, or what we call aged leaves. Those ones that have changed the color. Those ones that are no longer green. They also make the petals to have certain colors. For example, there is a carotenoid that they call xanthophyll. It makes structures of plants yellow, and then we have carotene to make structures of plants orange. If you know carrot, the real carrot that you eat, it's made of carotene, and this carotene is the one that actually is modified to form vitamin A in your what? In your eyes. And it's the one that is used to synthesize or make the structure of the retina. And you know the retina is that guy in your eye that gives you the capacity to see. So if you don't eat enough fruits that contain carotene, you are likely to have poor vision because of the less retinine in your eyes. So now we have looked at the types of pigments, but where do we get the evidence that actually these pigments are the ones that trap the solar radiation that is used in photosynthesis? The evidence comes from what we call an absorption spectrum and an action spectrum. An absorption spectrum is a curve that shows the relative amounts of a certain wavelength of an electromagnetic radiation absorbed by a given pigment. Now, I want first of all to talk about the word relative. When we say relative, we don't mean like if I tell you to go and to the shop and you buy 10 kilograms of rice and then you go and buy them. There we say that you have bought that by saying 10 kilograms, that's an absolute quantity of rice. But if I go and tell you on those 10 kilograms of rice you've bought, take 20%, it doesn't mean that 20% you have to take 20 kilograms. Simply it means get a portion. So you are comparing, relative is like comparing one parameter with another. For example, here when we say the relative amounts of a certain wavelength, if you have like your wavelength of light, if like 20% is absorbed, it means that's what we mean by relative. You can't measure this wave, the amount of this wavelength. You can't get maybe you can't measure it. You can't measure its absolute amount, but you can measure the percentage. So that's what we mean, or the ratio which has been taken. Are we together? So that's what we mean by relative. So you get a certain wavelength of light. Let's say you get yellow light. Yellow light has its own wavelength in nanometers, and you strike it towards a certain pigment, and then that pigment will absorb like 20% of that wavelength, of which it is all the light is electromagnetic. It has both magnetic and electric properties, so we call it electromagnetic. So the percentage of that wavelength that is absorbed by this pigment, the chlorophyll pigment that you have, is what we shall call. And if you have this pigment, because you have like your light ranges from the visible light that will be absorbed it begins from violet and up to red, but usually we include ultraviolet and the infrared, because plants can absorb that wavelength, even if for us we can't see it, but at least they can absorb some. So now you can see that from the ultraviolet has the highest energy in our visible spectrum. Ultraviolet has the highest energy. Then when you move from ultraviolet, violet, blue, green, yellow, orange, red, then you go to infrared. So infrared has the lowest energy. So now as you begin experimenting with ultraviolet, then you go to violet, then you go to blue. So you keep on noting down how much has been absorbed, how much has been absorbed, how much has been absorbed. Now you draw that graph together, and then you plot. Depending on here down, you put on the horizontal axis, you put the amount of, you put the wavelength of the light that you are dealing with. Each color has its own specific wavelength. Then on the vertical scale, you put the relative amounts that have been absorbed, and then you plot. So the curve that you will get is called an absorption spectrum. It shows the relative amounts of a certain wavelength of an electromagnetic radiation absorbed by a given pigment. This pigment can be chlorophyll A, it can be chlorophyll B, it can be chlorophyll C. It can also be a carotenoid, as we have seen. Hope you've understood that. Then we get to what we call an action spectrum. An action spectrum is a curve that shows the effectiveness of a certain wavelength of an electromagnetic radiation in stimulating a given process. So which process are we talking about? There are so many processes. It can be germination, it can be flowering, it can be photosynthesis. So in this case, we are talking about what? Photosynthesis. Now, after you plotting an absorption spectrum, of course, when this light is absorbed, it will not just be used for nothing. It will not be, it won't be wasted. It must be used for something because this light contains energy. So this energy is going to be given to these pigment molecules. And these pigment molecules, of course, chemical reactions will take place and a certain process will be initiated. Now, how much effective or how good or how best has this absorbed radiation, how much has it influenced a certain process? For example, if you look at photosynthesis, if you give a plant ultraviolet light, what is the rate at which photosynthesis takes place? How effective is this spectrum, or is this specific wavelength in stimulating photosynthesis? Does it stimulate it higher also, or there is no photosynthesis that takes place at all? So you can measure this kind of graph. You can measure this. You can get the data to plot this kind of graph by doing this. For example, you can give a plant, let's say you begin with violet light. You give the plant violet, it absorbs, and then when it absorbs, for you, your target is not to measure how much it has been absorbed because you did that in the absorption spectrum. But now your target is what is the rate of photosynthesis? How much photosynthesis is taking place? So how do you measure the rate of photosynthesis? You can measure the rate of photosynthesis by how much oxygen is being given out. Right together? Because it's very difficult to measure how much carbon dioxide is being used up, but you can measure how much oxygen is being given out. Of course, there are techniques for measuring that, the amount of oxygen that is being given out. Now, you find that after some time you get a steady rate of photosynthesis and a specific wavelength, let's say the violet wavelength, and then you record that all together. Say this is how much, this is the rate at which, of course, you get the amount of oxygen produced divided by the time it took, and then you get the rate, and then you note that. Then you go to another wavelength, maybe blue. Then you also record. You see how much oxygen is being produced over which period of time you divide, and then you get the rate. Like that until you reach red or even infrared. Then you'll still plot a kind of graph. But you should be knowing which pigment you are dealing with. Is it a carotenoid? Is it a chlorophyll A? Is it chlorophyll B? Is it chlorophyll C? Depends on what you want to find out. Then you plot that kind of graph. So that will be the action spectrum. Now, I was telling you that these two are actually evidence that these pigments are actually involved in in the process of photosynthesis. Why? Because when you plot these two graphs, they are two separate graphs. But if you plot them, their shape turns out to be very similar. Right together? That means the more of a certain wavelength that is absorbed by a certain pigment, the higher the rate of a certain process. Right together? And the less the lesser the amount of a certain wavelength that is absorbed, and that will be the slower by which a certain process will be stimulated. Right together? So since they turn out to have a similar shape, that means actually the chlorophyll and what, they are actually involved in the process of photosynthesis. So this is a combined absorption and action spectra. So you can see that's what I was talking about. This is now the relative absorbance. I forgot to put here action. Absorbance is for this absorption spectrum. Then up here there must be action spectrum, the effectiveness of the process. So you can see here, at first we begin from low, a bit low, certain point low, then we increase, then it decreases, increases, and then it declines, then it increases, then it declines. So from 400 nanometers to 500 to about 450 nanometers, basically from 350 to 450, there is an increase, rapid increase actually. Then from 450 to about 500 or 510, there is a rapid decline. And then from 510 to about 600, there is a gradual increase. Then from 600 to about 650, there is a gradual decline, and then followed by a slight increase. Then from 650 to 600, about let's say 70, there is a very rapid increase in the rate of the amount of absorption, the rate of absorption. Then from 670 to around 700, we have a rapid decline or decrease in the absorbance of the chlorophyll molecule. Then you can see that that one is almost followed by this, the rate of what? Of photosynthesis, the effectiveness of the particular wavelength. You see that where there is maximum absorbance, there is maximum photosynthesis. Where there is minimum absorbance, there is minimum photosynthesis. Like that, like that, like that. So that's the evidence. In case someone asked you, that's what you could present. So now we are going to look at the mechanism of photosynthesis itself. How does photosynthesis take place? That's the question that we are trying to answer. Generally, it is known that photosynthesis will lead to the formation of carbohydrates. And there is a general equation which was suggested a long time ago, whereby carbon dioxide plus hydrogen plus water, you get carbohydrates, plus oxygen. I didn't finish the equation, but what matters is this. So in the mechanism, as we are trying to look for the mechanism of photosynthesis, I want you to know that photosynthesis actually takes place in phases, and there are two phases of photosynthesis. The first phase is what we call the photochemical phase. You can also call it the light-dependent phase. Photochemical, that means it involves light and some chemical reactions. So since it involves light, we also call it a light-dependent what? Phase. Then you have the biosynthetic phase. That means it involves some kind of biology, which is synthesis of things basing on biology, not mechanically. So it's biosynthetic phase. We also call it the light-independent phase. However, the light-independent phase and light-dependent phase are the two commonly used names. But in case you come across these two, the photochemical and biosynthetic, don't say the teacher never mentioned them. So light-dependent phase. I want to first of all make this clear. When we say light-dependent and light-independent, it doesn't mean that strictly the other one cannot take place when light is there. No. It actually means that it can take place when light is there or not. Are we together? But simply, it does not need light. It's like, should I say, how should I say? The light-independent stage or phase, for it can take place whether in darkness or in light. But it does not need light for it to take place. It's not that when the light is not there, it will not take place. No, it can take place whether it's there or not. So we say, in other words, it is actually independent. Then the light-dependent phase, for it strictly, it cannot take place unless there is light. Are we together? But even though the other one, we call it the light-independent phase, I want you to know that today, our focus today is going to be on the light-dependent phase. But even though the other one is called the light-independent phase, I want you to know that it depends on the light-dependent phase. The products of the light-dependent phase are the ones that go in the light-independent phase. Are we together? So that means if by somehow the light-dependent phase gets some problems, that means even the light-independent phase will not take place. I hope you've got it. For example, ATP and NADPH, these are molecules which are synthesized in the light-dependent phase, and they are the ones that go in the light-independent phase. It's like the way you see cows. When cows are eating food, they first of all go around and gather everything during the day when there is light there. They go and gather everything, eat and store in their stomach. Then afterwards they will come back in their kraal when they are resting, and they begin to regurgitate and chew that food again. So we can say that regurgitation, we can call it the light independent phase. And then the other gathering of food and grazing, we can call it the light dependent phase, of which regurgitation can take place where there is light or whether there is no light. That chewing you see in cows at night. Where there is light, where there is no light, they can do their thing. But they depend on the other light independent phase, where they have to first go and gather during the day. They first have to go and gather whatever they will eat. Then they will come back, rest, and then begin to process the food slowly. So this is the same thing applies here. Hope you've understood that. So basically, in our light-dependent phase, we have two things, the major targets. We have synthesis of ATP and synthesis of NADPH. What is NADPH? I'm sure you looked at NADPH. You looked at it in Chemicals of Life. These are coenzymes. Right together, these are coenzymes which carry, for example, most of the time they carry hydrogen, and then they can transfer. They can they can act as stores of hydrogen. This hydrogen can be used to reduce other compounds. You know from chemistry that when you add hydrogen to any compound, you have reduced it. You know, we have three types of reduction. You can reduce by giving, you can reduce a compound or any substance by giving it electrons. You can also reduce it by removing oxygen from it. You can also reduce it by giving it hydrogen. Then oxidation also is in three parts. You can reduce something. You can oxidize something by giving it oxygen. You can oxidize it by removing electrons. You can also oxidize it by removing hydrogen. So, when this NADPH donates its hydrogen to any compound, that process will be a reduction process. If this NADPH removes hydrogen, if this NADP removes hydrogen from any compound, we shall say that compound has been oxidized. So we have trapping of protons of light by photosystems, the things which take place here. We shall see what a photosystem is. So we trap light by photosystems, and then we produce ATP. We also produce NADPH. Then we split water to form oxygen as a byproduct, and of course hydrogen ions, which will go to this NADP to form NADPH, and also some electrons, which we shall see their use later. So, what is a photosystem? A photosystem, basically, I want you to know that in the leaf we have chloroplasts. We have cells, and then in the cells we have chloroplasts. Then in the chloroplasts we have what we have called the thylakoids. So on the surface of these thylakoid membranes, we have groups of chlorophyll molecules, right together, or pigment molecules. These groups are arranged in a way that they are like an antenna. If you have a TV at home, you know how an antenna, what the role of an antenna. So we have always a central atom in this antenna of pigment molecules in on a thylakoid membrane. We have, you can check out those pictures in chapter 7 of biological science. We have a central molecule, which is always chlorophyll A. Are together? A central molecule, chlorophyll A, surrounded by other chlorophyll molecules. They can be chlorophyll B or chlorophyll C. Then also surrounded by carotenoids. Are together? So the carotenoids are on top of chlorophyll. Then the chlorophyll B or C, they are on top of chlorophyll A. So chlorophyll A is always in the what? In the center. So that arrangement is like that in order to maximize the amount of solar radiation that can be absorbed, and which will always, of course, the amount of solar radiation absorbed will determine how much energy or how much ATP will be produced during what? Photophosphorylation. We have seen that one of the major aims of the light-dependent phase is to make ATP, and that ATP is made during through a process known as photophosphorylation. We are going to see what photophosphorylation means. So, but basically that's what a photosystem is. Simply an arrangement of what? Carotenoids and chlorophyll molecules, which maximize the amount of what? Solar radiation absorbed, which is subsequently converted to ATP through photophosphorylation. And I've told you, in a photosystem, we always have chlorophyll A in the center, chlorophyll B surrounding, or chlorophyll C, and then you have carotenoids covering, protecting, in other words, chlorophyll A and chlorophyll B are together. So that arrangement is always, the aim of that arrangement is always to maximize or to increase the amount of solar radiation that can be absorbed, and technically converted into ATP. So that's the composition of photosystems. So what happens is that when these photosystems are, every photosystem, the funny thing is that the carotenoids and chlorophyll B or C, for them, their role is to trap the light, the photons of light. You see, light can be regarded as a wave or matter. When we say matter, that means it has particles. So when we say it has particles, those particles, the smallest particles of light, are called photons, and each photon has a certain amount of energy. So when these chlorophylls, chlorophyll A, chlorophyll B, but beginning with carotenoids and chlorophyll B or C, for them, when they absorb this energy, they simply hand it over to the central chlorophyll A molecule. Are we together? So since for them they don't do anything more than that, they simply help chlorophyll A in the center to access more energy. We call them accessory pigments. And you can see that carotenoids and chlorophyll B or C, for them, we call them accessory pigments. Then chlorophyll A, for it where chlorophyll A is, we call it the reaction what? Center. So I think I've explained that. So you can see that. However, now when it comes to, we have two types of photosystems. We have photosystem 1 and photosystem 2. Why do we say that we have photosystem 1 and photosystem 2? Now that one depends on the wavelength of light that the central chlorophyll A atom can absorb. I told you light can be regarded as a wave or matter. So when we consider light as a wave, each wavelength has, each color, let's say yellow, green, blue, violet, indigo, what, each of them has a certain wavelength. Are together? But we have two types of chlorophylls. A, that is chlorophyll A, the central ones. One type of chlorophyll A absorbs a wavelength of 680. That is towards this is towards green. I mean, this is towards red, and we call it pigment 680. Why? Because the wavelength of light it absorbs is 680 nanometers. Nanometers, those are the way you can say meters, then you say centimeters, then you say millimeters, then you say micrometers, then you say nanometers. So nanometers are the units we use in the wavelength of what? Of light. Then we also have another pigment, still chlorophyll A, which absorbs the wavelength of 700 what? Nanometers. This one is also, as I've explained, still we represent them in nanometers and stuff. So depending on the amount or the wavelength of light absorbed by a specific chlorophyll A molecule, we have photosystem 1 and photosystem 2. Photosystem 1 it absorbs wavelength of 700 nanometers. Photosystem 2 absorbs wavelength of 600. There was a certain typing error here. It is supposed to be 680, not 860 nanometers. So this is photosystem 2, photosystem 1. However, now why do we call them photosystem 1, photosystem 2? We have seen why we call them this one. We have seen why we call it pigment 680 and pigment 700, because of the specific wavelength that they absorb. But why do we have photosystem 1, photosystem 2? Photosystem 1 was the first type of photosystem to be discovered. Then photosystem 2 was the second to be discovered, and they have not discovered any other. So that means we only have 2 photosystems. Now, what is photophosphorylation? Basically, I told you one of the major reasons why we have the light-dependent stages in order to synthesize ATP. Are we together? So now the word photo means light. Phosphorylation means adding a phosphate group to a certain compound. Now, photophosphorylation simply means making ATP from ADP and inorganic phosphate, of which ADP, you know, I'm sure you know what ADP means. Adenosine diphosphate. So types of photophosphorylation. We have cyclic photophosphorylation and non-cyclic photophosphorylation. However, I want you to know that even if we are calling them photophosphorylations, there is also synthesis of NADPH. Are we together? We are calling them photophosphorylations, but in one of these photophosphorylations, at least there is synthesis of NADPH. And you know, the targets of the light-dependent stage is to make ATP and NADPH. So let's look at these types of what? Photophosphorylations. So let's begin with the cyclic. This is the most primitive type of photophosphorylation. It is found only in prokaryotes. Are we together? Those prokaryotes that are so primitive, the bacteria, that's where we find this cyclic photophosphorylation. It is ancient, it is of a long time ago. However, I think it is the origin of the name photophosphorylation. It is where ATP synthesis occurs, but there is no synthesis of NADPH. And I told you, the moment you tamper with the light-dependent stage, that means even the light-independent stage will not occur. Are we together? So, here, ATP is made, but there is no NADPH synthesized. That means the other light-independent stage will not occur the way it occurs in other organisms. Are together? Because there is no NADPH. Then, so why do we call it cyclic? An electron moves cyclically through electron carriers. You see, in the thylakoid membrane, we have molecules there. We have enzymes, we have a lot of things there. So some of them are called electron what? Carriers. Why? Because they transfer an electron from one molecule to another. So in that case, they become electron what? Carriers. So however, in cyclic photophosphorylation, a chlorophyll molecule will lose an electron. Let's say chlorophyll A will lose an electron. When it loses an electron, that electron will go into electron acceptors and then move cyclically, move through a series of electron carriers, and then it will come back to the chlorophyll molecule from which it came. So that means, in other words, it has moved in a circle. When you move, no matter how long the distance you have moved, but as long as you go back to the same point, that means you have moved in a circular pattern. So, funny enough, only one photosystem is involved, and that is photosystem 1. Are together? So photosystem 1 is where we have the chlorophyll A molecule, the one that absorbs 700 what? Nanometers. That's where we have cyclic photophosphorylation. Are together? I told you it was the first to be discovered. Photosystem 1, the one of P700, it was the first one to be discovered. And that is why it is even found in the most primitive type of what? Organisms, the bacteria and stuff. Now we go to the non-cyclic photophosphorylation. What should we know about this? We know that this one is actually found in advanced organisms, and ATP is synthesized. Even NADPH is also synthesized. There is production of oxygen or sulfur as a byproduct, because we shall see how this oxygen and sulfur comes about. But just know that wherever we have NADPH, just know that either oxygen or sulfur was produced as a byproduct. It involves both photosystems, photosystem 1 and photosystem 2. It is found in plants, protists, and some bacteria. That one, the point of bacteria depends which type of what? Bacteria. Some bacteria, the primitive ones, have the other type that we have seen, the cyclic. And then here, these ones have, these are the bacteria we find here. For them, they have this type of photophosphorylation. The reason why we call it non-cyclic is because once the chlorophyll A molecule loses an electron, that electron will go for good. It will never come back to that photosystem, to that chlorophyll molecule again. Are you together? What will happen is that maybe because you must fill up that gap. Where you have lost an electron, you must replace it, but you don't replace it with the one that exactly you lost. The electron that will fill up this gap will come from water or hydrogen sulfide, from what we call the Hill reaction. That's where you split water to form oxygen gas, hydrogen ions, and electrons. The electrons will go up and fill up the lost electrons. We are going to see that scheme, but even in BS it is there. It is the most advanced type of photophosphorylation. We saw that. So this is the illustration of cyclic photophosphorylation. Here we see a photon of light hits or strikes photosystem 1, where we have the pigment 700, the one that absorbs 700 nanometers. But I want you to know this is a photosystem. It's not just made up of only this pigment. There are so many carotenoids around, so many chlorophyll A or chlorophyll B, and then in the center we have this P700. That's why we call it photosystem 1. So when an electron from pigment 700 will move, will be, when this chlorophyll molecule, when light strikes this chlorophyll molecule, the P700, it absorbs energy. This energy boosts these electrons to higher energy levels. If you remember your senior 2 chemistry very well, you know that we have atoms, and atoms have electrons, and those electrons are in shells. So we have shell 1, the one closest to the nucleus, and then we even have those ones that are further from the nucleus, the ones that the one that we, for example, the outermost shell, it is always further from the nucleus. So I think you have studied in chemistry, right, so far, that those shells that are far away from the nucleus have a higher energy level compared to those ones that are closer to the nucleus. So when the photosystem absorbs light, first of all is the carotenoids and the chlorophyll B or C molecules. For them, they absorb the light, as we saw, they hand it over to what? They absorb the energy, they hand it over to the central atom, central chlorophyll molecule, which is P700 in this case. So, plus its own energy that it absorbs, the 700 nanometers, it will do what? It will absorb enough energy. This energy can be used to move some electrons from those energy levels which are closer to the nucleus of the atoms to higher energy levels. When they move to the higher energy levels, these electrons, in case the energy is sufficient enough, it can even be lost from the atom completely, and then that means you form an ion. And when you lose an electron, the atom will become what? Positively charged. Are you together? When they become positively charged, that means that atom is found in chlorophyll. That means our chlorophyll has been oxidized, because I told you one of the ways of oxidation is losing an electron. So when you lose this electron, it doesn't go in space. It is picked up by what we call an electron acceptor, and this is called ferredoxin. F-E-R-R-E, then doxin, ferredoxin. Now, ferredoxin picks up this electron. Then it passes it over to a series. There are so many electron carriers here. Are you together? So many electron carriers, but I just showed a few of which these are the only ones that you need. You don't need a lot of them. So first of all, we shall give it to plastoquinone, then to cytochrome. But I want you to note that as the electron is being given to other electron carriers, we are moving downwards. This downward movement represents that the electron is losing energy along the way as it is moving, because it picked up high energy and then it even reached an extent it couldn't even be contained. So now as it is moving through a series of electron carriers, it loses energy. When it loses the energy, this energy it loses is used to make ATP from ADP and inorganic phosphate. Now this electron from cytochrome, these cytochromes are basically they basically contain copper, those ions, those transition metal ions. So you shouldn't so much, they contain proteins and metals, so you shouldn't focus so much on them, but just know the name of cytochrome and then plastoquinone. So from there, this electron will eventually go back. Remember, it left this guy when it is positively charged and lacking one electron. So then this very electron goes back to fill up that gap. Are together? So that's all with cyclic photophosphorylation. You can make some more reading about it. Now we come to the guy, non-cyclic photophosphorylation. This is the one that we find in plants, by the way, and most of the photosynthetic guys. I told you the other one is primitive. Now here, and I think you've seen that here, only one photosystem is involved. That is photosystem 1. Now, here we have 2 photosystems. The first one is photosystem 2, the one of 680 nanometers. and then photosystem 1, the second one. Now, why am I saying it's photosystem 1 and I'm calling it the second one? I told you this was the first one to be discovered. The second one. It was the first to be discovered. That's why they call it photosystem 1. But as far as the arrangement is concerned of non-cyclic photophosphorylations, it comes second after this one. The one that was discovered last, in terms of in the order or the chain or arrangement, it comes first. So don't confuse those two things. So we begin with photosystem 2. As I told you how energy, the other guys, the carotenoids, get the energy, then they will transfer it to the neighboring chlorophyll B or C molecules, then that energy is now channeled to the center, where we have now chlorophyll A, which will absorb the wavelength of 608 nanometers. So this guy has its first electron acceptor as plastoquinone. Then from plastoquinone, it hands it over to a complex. A complex, sorry, is a protein, and with some other substances, can be elements like atoms, as in metals. As we call it, a complex, something which is composed of so many parts, not just one part. Then we go to plastocyanine. This one contains copper, and then from there we shall have pigment 700 or photosystem 1. Photosystem 1, the same thing will happen. The electrons will move to ferredoxin, as we saw earlier, then to a series of what until we get NADP reductase. NADP reductase, that means you are reducing NADP by giving it hydrogen ions to form NADPH. Where do these hydrogen ions come from? I told you of the Hill reaction, where water splits. Hill reaction simply requires the presence of light. Here on the arrow there must be light to indicate that the light which is there splits the water, of course with some enzymes. You can't split water like that. With some enzymes, then that water we shall get hydrogen ions, we shall get oxygen gas, and then 4 electrons. These electrons will come and fill up the gap of the electrons which were lost. Altogether, we lost some electrons here in pigment 780. So these electrons from water will come and fill up the gap. Then the same thing will happen. Light will strike this photosystem 2. Electrons will move to higher energy states. They will be picked up by plastoquinone. Plastoquinone transfer them to a complex which we call B6F, and then, but you can see that they are moving. The electron is moving downwards in terms of what? Energy. As it is moving downwards, it is losing energy slowly. Now this energy is used to synthesize ATP. Right together, as it is moving down, it is synthesizing what? ATP. Now, from then eventually the electron will lose enough energy, and it will now be picked up by photosystem 1. Still, since this is a photosystem, still it will pick up light, it will absorb light, and then it will boost another what? Electron. So the electron will move. So the electron that comes from pigment 700 or photosystem 1 is replaced by the electron that comes from pigment, or the electron that is lost from photosystem 1 is replaced by an electron from photosystem 2. An electron lost from photosystem 2 is replaced by an electron from the splitting of water, what we call the Hill reaction. And then eventually the splitting of water will result into oxygen, electrons, and hydrogen ions. I've already told you where the electrons will go. So the hydrogen ions will combine with oxidized NADP. You can see this NADP, it's oxidized. And then it will combine with it. By saying oxidized, I mean it does not contain any hydrogen. Right together? So when you give it hydrogen, when you add their hydrogen, that means you are reducing it. And when you reduce it, you form reduced NADP, or what we call NADPH. This is supposed to be capital A, NADPH. And then that's what we call non-cyclic photophosphorylation. We call it non-cyclic because technically electrons are moving in one direction. They don't come back to the original source where they came from. Are together? And then you can see that in these non-cyclic photophosphorylations, both ATP and NADPH will be made. So next lesson we shall see what comes later. But the moment you exceed here, then you have entered the other phase, which we call the light independent what? Phase. I would like to conclude from here. Hope you'll enjoy. Where you've not understood, you'll contact me in our Google class. You tell me where you've not understood, and I'll try to explain.